They work with a mentor, and they investigate something that they're interested in. So without further ado, I'd like to introduce Kate Priest speaking about future applications and ethical concerns of CRISPR genome editing.
So hello everyone. Like Dr. Cook said, I'm going to be talking about CRISPR today and the ways it can be applied in gene editing and some concerns that people have with that. So I've got a lot of material to cover, so I'm just going to get right into it. CRISPR is short for clustered, regularly interspaced, short palindromic repeats, and it functions as a form of prokaryotic adaptive immunity. It's basically their immune system. So it works by incorporating a portion of an invading pathogen's DNA into its cellular memory, then generating strands of guide RNA based off of that, and loading that RNA onto a Cas9 protein. It will seek out matching DNA in future pathogens and cut the DNA at that area. With their DNA cut, they struggle or are completely unable to repair it, which means the pathogen will die. So I have a little video here that's just going to go over it real quick so that you understand better what I'm talking about, because I'm not very good at explaining it.
Great ideas can come from anywhere.
First, a heartwarming moment.
This doesn't count toward my time, right?
Totally good.
I don't know how to skip this.
I don't think you can.
Oh,
You probably
then zoom in and dictate the mood and vibe. And when it all comes together, launch your idea into the world where it belongs. It's your business, your website. You do it yourself.
In a document, if we suspect we've misspelled a word, we can use the find function to highlight the error and correct it or delete it. Within our DNA, that function is taken on by a system called CRISPR-Cas9. CRISPR is short for clustered, regularly interspaced short palindromic repeats. CRISPR consists of two components, the Cas9 protein that can cut DNA and a guide RNA that can recognize the sequence of DNA to be edited. To use CRISPR-Cas9, scientists first identify the sequence of the human genome that's causing a health problem. Then they create a specific guide RNA to recognize that particular stretch of A's, T's, G's, and C's in the DNA. The guide RNA is attached to the DNA cutting enzyme Cas9, and then this complex is introduced to the target cells. It locates the target letter sequence and cuts the DNA. At that point, scientists can then edit the existing genome by either modifying, deleting, or inserting new sequences. It effectively makes CRISPR-Cas9 a cut and paste tool for DNA editing. In the future, scientists hope to use CRISPR-Cas9 to develop critical advances in patient care or even cure lifelong inherited diseases.
Okay, so that's basically how CRISPR works, and so the reason why I'm talking about this today is because CRISPR is a rapidly expanding field, and I think it's going to be something that's going to be in our lives a lot more going forward. But because there are some concerns about its use, it can basically edit any and all genes. Well, not all, but most genes. There are some people worried about the implications of that kind of thing. So CRISPR regulation might be something you have to vote on in the future. It might be something you might receive a CRISPR-based treatment in the future, or your life could be impacted in other ways by CRISPR. So I think it's important for everyone to know its abilities and also the implications of its use. So first I'm going to go over some future applications of CRISPR. I'm going to begin by talking about nutrition. So one way we've been using CRISPR is to produce gluten-free wheat. So celiac disease is something that affects about 1 to 2% of the population. That's pretty common, but despite that, there isn't a lot of available gluten-free food for people afflicted by celiac disease, and about 50% of patients do not follow a strict celiac disease diet because of lack of options. And also, the paper I was reading said the appalling palatability, which I thought was funny. I've tried gluten-free food. I don't think it's very good, so I had to agree. But researchers have been working on editing the kind of wheat we use, which is Triticum aesthetivum, to remove the genes that cause celiac disease. So this kind of wheat is called an allohexaploid, which means it's actually several strains of wheat combined with a total of 3 genomes and 6 sets of chromosomes. So the 3 genomes are AA, BB, and DD, and the D genome, which comes from a wild strain of wheat, improves the wheat's bread-making abilities, but also increases the amount of immunological peptides, which are basically what causes that immune system response to the wheat. The allergic reaction to this wheat is what we call celiac disease. And these peptides that are causing this are found in 2 different proteins that generate gluten. They're called gliadin and glutenin. Most of the peptide groups can be found in alpha-gliadin. So researchers have been working on knocking out the alpha-gliadin genes to reduce the amount of immunological peptides being produced. And the result was an 85% reduction in the gluten protein, which rendered the wheat much more palatable for celiac disease patients. Another way CRISPR has been used on food is making vitamin-enriched tomatoes. They have been working on infusing them with vitamin D to give them greater health benefits. Lack of vitamin D can lead to higher cancer, higher dementia, and higher COVID-19 risks. So it's important to get a healthy dose of vitamin D, and most of that is going to be coming from your food. You get some from the sun, but most of it is from your food. There's a lot of vitamin D in tomato leaves, but that's not something people usually eat. So scientists have been working on incorporating vitamin D into the actual fruit for consumption. So they do that by turning off the S17DR2 enzyme in the tomato, which is what makes the molecule 7-DHC into other things. If left by itself, the molecule 7-DHC will be converted into vitamin D3 when exposed to sunlight. So by turning off the enzyme that converts it into other things, that molecule will begin to accumulate in the tomato, and then when exposed to UV light, it'll be converted into a high amount of vitamin D3. When they did this, the resulting amount in the tomatoes was equivalent to 2 hard-boiled eggs, which is quite a significant improvement. Moving on, it's also used to treat disease. One of the most well-known applications is in treating sickle cell anemia. Sickle cell anemia is a genetic disorder. It's a mutation in gene HBB, which codes the beta subunit of adult hemoglobin. The result is rigid, sickle-shaped blood cells that are like sticky. They form clumps in your blood vessels, which can lead to clots. This can cause hemolysis, inflammation, blood clots, and which will eventually lead to chronic anemia, organ pain, organ damage, and an early death from blood clots in the heart or brain. So these symptoms don't manifest until the baby is born and adult hemoglobin starts to be produced. Fetal hemoglobin does not have the sickle cell disease. So one of the ways scientists have been working on treating sickle cell is by trying to return some of that hemoglobin to the adult body. So they did this by taking healthy CD34+ donor cells. Those are bone marrow cells. That's what's producing your red blood cells. and edited it using a selected guide RNA. They used an RNA that produced the highest amount of fetal hemoglobin. Then they used a CRISPR system they designed called OTQ923 and injected it into the patients. This system knocks out the gene suppressing the production of fetal hemoglobin, which allows the body to begin producing it again. By the end of it, all of the patients had 19 to 28 and 26.8% of their total hemoglobin levels being composed of fetal hemoglobin, and their symptoms drastically decreased. Another disease that's being treated is transthyretin amyloid cardiomyopathy. This is when TTR proteins, which are responsible for transporting thyroxine and vitamin A, misfold, clump together, and get stuck in the heart tissue. The heart becomes less flexible, which losing its ability to contract. This is a problem because if the heart can't contract properly, it can't pump blood properly. You'll be getting less oxygen to your tissues. That includes your heart, so your heart is going to suffer too. This can lead to congestive heart failure, conduction system disease, cardiac arrhythmias, and extracardiac manifestations. So what they did was they used an LMP that's basically like a lipid vesicle that can pass through phospholipid bilayers, and they used a custom CRISPR-Cas9 system inside it, and they injected this LMP into patients. So the patients who received 0.1 milligrams per kilograms of the LNP had a reduction of 52% of the TTR protein, while those who received 0.3 milligram per kilogram LNP had a reduction of 87%. So this also reduced their symptoms a lot because it was reducing the amount of the protein that was causing all the problems in the first place. So we can also use CRISPR for modifying species. One of the examples are in mosquitoes. Anopheles gambiae is a form of mosquito that causes malaria. Scientists have been using something called a gene drive, where they basically combine the concept of trait acquisition with a Cas9 system, a CRISPR-Cas9 system, to spread the trait through the population. Because CRISPR moves extremely fast, much faster than other gene editing techniques, this is a very effective way to alter a gene very quickly. I'll get into more why later in my presentation. Anopheles gambiae is the type of mosquito responsible for malaria, like I said. Within their genome is the gene double sex, or DSX, that determines the sex of the mosquito. There are two forms, AGDSX male and AGDSX female, and each have some crucial differences. The one I'm going to focus on here is that AGDSX female has an exon, that's a gene sequence used to code proteins, that is present through every species of Anopheles mosquito. And researchers were able to use a CRISPR-Cas9 system to disrupt the boundary between intron 4 and exon 5, that exon that only the female mosquitoes are using. And they disrupted the boundary between it, which was causing an error in phenotype manifestation. Because only the females use exon 5, the male mosquitoes were unaffected, while the females, any of them with the homozygous edited allele, were born with an intersex phenotype, which rendered them sterile. Within 7 to 11 generations of reproduction, 100% of the mosquitoes had been rendered sterile. That's about 77 to 144 days. So they completely wiped out this population, basically, within a third of a year. Another way you can use CRISPR for species modification is to actually bring back extinct species. So something scientists have been working on is trying to restore the woolly mammoth. They believe that this could help in saving endangered elephant populations, as well as restore environments that are deteriorating. What they do is, it's kind of like what they do in Jurassic Park. They extract DNA from fossils and they read the genome. Then they compare that genome to existing genomes of similar animals, and then they look at the differences and edit the genome of the current animal to have genes that match the extinct animal. So in terms of woolly mammoths, there are millions of differences, so they're not doing them all, but they can edit the phenotype. That's basically the phenotype genes. Those are basically the genes that are expressing like the physical characteristics. And basically to give it, they can give it longer hair, they can give it larger tusks, bigger stature, that kind of thing. So it wouldn't be a 100% identical copy of a woolly mammoth, but it would be very close. And then the last thing I'm going to talk about in terms of application is camera, which is a form of cell recording. So camera is short for CRISPR-mediated analog multi-event recording apparatus, and it is a tool that can record events in a cell's life. So it's basically a modification of the CRISPR system. It's modified to trigger. CRISPR is modified to trigger only after the cell experiences a certain stimulus. When this occurs, there's two different forms of camera that can be triggered depending on which one scientists are using. Camera 1 uses several round DNA sequences called plasmids. When the stimulus occurs, an RNA plasmid is generated. It combines with the Cas9 protein and seeks out two other plasmids that are in a certain portion of the DNA. The CRISPR system is going to cleave one of those plasmids and leave the other one untouched. Then they can then compare the ratio of the cleaved plasmids, or plasmid 1, to the uncleaved plasmids, or plasmid 2, to tell if a stimulus has been presented, how long it's been present, and so on. Camera 2 does not use extra plasmids. Instead, the guide RNA will swap out nucleotides at certain locations called safe harbors. Basically, it's called a safe harbor because it can edit that gene without causing any adverse effects to the organism. It'll swap out cytosine for thymine and guanine for adenine. And researchers can look for these nucleotide changes to determine the presence of a stimuli. So those are some applications of CRISPR. I think it's all very exciting, the things we're able to do with it, but it's not without its flaws, and there are a lot of concerns surrounding this tool, which is what I'm going to get into now. The first issue is something I want y'all to keep in mind when I talk about the rest of the issues, because they kind of stack, and that is that CRISPR is still pretty unpredictable. Mistakes are still frequent. Sometimes when trying to edit one gene, they accidentally hit another gene instead, or another gene along with it, which can cause other problems when that gene isn't being expressed properly. There was a case where, this wasn't using CRISPR, but a different form of gene editing, where they edited a boy's genes, they hit the wrong one, and he died from it. So it isn't a perfect system yet. Another problem is CRISPR mutations are still difficult to track. Pretty much impossible, in fact. I mean, once you're editing out a gene, you're not really leaving any trace behind, which can be a problem for a lot of reasons. If there was an undesirable edit, you won't really know what changed to go back and fix it unless you have that in your research. You'll have to go all the way through and look at all of the genes again and compare them all to an unedited version to try and find what gene you fixed. What gene you changed. you can imagine this could be a really big problem if someone was, not to be like a conspiracy theorist, but to make a bioweapon. If like a virus was edited or something like that, it would be a bit tricky to find out what exactly to change to make that virus into a non-dangerous form. Another problem is that CRISPR, we don't really know how CRISPR edits will function in the future. We don't know if they're going to mutate or how they're going to mutate. So that's another thing to keep in mind. And then one more problem is that CRISPR is very, very cheap to use. I read, and I haven't checked back up on this, so I can't say for certain. I think it's around $38 for to harness CRISPR in your lab. Obviously, you need other things in there too. You need stuff to sequence RNA. You need donors for cells sometimes. But CRISPR itself, the system is very inexpensive, which means pretty much anyone can get their hands on it, which can be good for research, but it can also lead to problems, which is what I'm going to get into. Here are some examples. One of the problems people encountered when using CRISPR is that they were using it on subjects that could not consent. And by that I mean embryos. This first started with the scientists at Sun Yat-sen University who used non-viable embryos to experiment with CRISPR editing. A non-viable embryo cannot be born, just for the record. It was fertilized by 2 sperms, which renders it unable to develop into a fetus. They were using the embryos to experiment with editing human beta globin. They were basically trying to refine their targeting techniques to not be hitting other genes accidentally, but their work still sparked controversy because that was still humans they were working on. And so their work was rejected by most of the top science papers based on ethical grounds. It got worse, though. Another scientist named Jiankui He revealed that he had used CRISPR-Cas9 on viable embryos. He was planning to make a CRISPR-modified baby. So he obtained the consent of several sets of parents, the males containing an HIV gene and the females not. He then attempted to modify the CCR5 gene, which is responsible for HIV, in the fetuses. According to him, the gene was inactivated. One of the babies, they were twins, one of the baby girls was born immune to HIV, although it's debated whether he actually did it or not, but he claims one of the babies was born immune to HIV. But the backlash was extreme. Like I said on the previous slide, there's a lot of problems that can go with CRISPR. He put these baby girls' lives in danger by editing their genes. We still don't really know how those genes are going to affect them in the future. One thing we do know now is that they are more prone to worse cases of West Nile virus and influenza because of the editing that their genes experienced. So another problem is eugenics. I know that some people think that eugenics isn't really a thing anymore, that we've gotten over it and that everybody recognizes it's wrong. I do not think that's true. I think that there is still a troubling amount of people who support it in some way or another, which I'm going to get into. That's a picture from the movie Gattaca, which is basically about a dystopian society where eugenics is used to decide like the hierarchy of people. So here I have some pictures of some cryobanks where you can purchase sperm. And if you look, you can see that you can filter out everything you want for your baby. You can pick the height, you can pick the race, you can pick the hair color, the eye color, the ancestry, you can pick the blood type. So you can see these people who are purchasing these, they can basically have like a designer baby, it's called. They can pick exactly how they want their baby's DNA from the donor to be. So you might think that that's only because they want their baby to look like them to match their family, but I don't think that's true. Over here I have an example. I have a graph showing the percentage of sperm donors, the demographics of sperm donors compared to the demographics in the U.S. population. Something that's important to mention is that you have to be approved to be a sperm donor. You can't just walk in and donate sperm. So these are the percentages of sperm donors who were approved, and you can see 74% of approved sperm donors, the pink is the donor, blue is demographic, were white, while 58.4% is the population of white people in the U.S. Over here, only 1.4% of sperm donors are black, even though they make up 13.70% of the population. Over here, only 0.4% were non-white Latinos, even though they make up 19.5% of the population. For Asians, it was 7.7 compared to 6.40%, and for people who identified as other, it was 16%. 16% of them were sperm donors, while they only make up 3.10% of the population. Another thing that you can observe is the education levels of sperm donors. You can see over here, only 2.3% of approved sperm donors were only high school graduates, even though they make up 27.9% of the population. Everyone else who was approved had a higher level of education. You can see over here is 8.3%, even though they make, for some college, even though they make up 14.9% of the population. These are starting to go up a little bit, but it doesn't actually surpass the population until you get to a bachelor's degree. That's when you get a whopping 70% of sperm donors that have a bachelor's degree, even though only 23.5% of the population does. And then I was supposed to fix that and I didn't. I didn't mean to write higher ed, I meant to write higher degree, but a degree higher than a bachelor's, so a master's or a doctorate, 13.10% of sperm donors are going to have that as compared to 14.4% of the U.S. population. So it's important to note here that it seems like they're trying to use education level to generalize intelligence, which is not how it works. Higher education does not correlate to higher intelligence, and historically there are certain demographics that have had less opportunities to pursue higher education, meaning that these demographics are being shut out from sperm donating because of something that isn't their fault. So to me, what this is saying is that even though eugenics is thought to be a thing of the past, we are still only letting certain types of people donate their genes to be passed on. People who are highly educated, people of certain races are getting more opportunities to pass on their genetics than people of other races or people of lower education. So I noticed this with egg donors as well. This article is was based off of a book called Eggnomics, in which a researcher interviewed different egg donors across the U.S. The bottom one is from an egg donation website, and something that stuck out to me was that it says, egg donors may also receive higher compensation if they have highly desirable characteristics, which again shows that they are picking and choosing what genes they consider desirable. So to me, this shows that we are not past the era of filtering out or uplifting different genes, which I consider very problematic. And how I think this applies to CRISPR is that if we as a society are already okay with picking and choosing genes, there is nothing to stop us from using CRISPR to do the same thing. If people are already paying to make sure their baby has blue eyes, then they're also going to pay to use CRISPR to modify their baby to have blue eyes, if that makes sense. I think really what bothers me is the education part of it all, because again, higher education does not mean higher intelligence. We don't know what causes intelligence. There's no one gene that causes it, so they're kind of just like crudely correlating these things, when in reality, education, how far you go, is based on a lot of factors, and intelligence is just one of them. So another thing that still occurs is forced sterilization, which is another age-old practice used in eugenics. So from the 1900s to the 1960s or 70s, sterilization was a very common thing, and it was mostly done to women of color and women who had or were accused of having mental illnesses. Although this is supposed to have gone away, as recent as 2013, which I know that's still like over 10 years away, but that's still pretty recent considering how far we thought we came. There were a lot of female inmates, prison inmates in California who were sterilized without their consent. The disproportionate amount of these women were women of color. So I think that's another thing that just shows there is still discrimination happening when it comes to passing on genes, and it just worries me that CRISPR will be used to harness this kind of stuff in the future. Moving on though, another problem is extinction. The previous forms of gene editing they're very slow to travel through a population because they go onto one chromosome. That means only like half of the organism's offspring can inherit it, while CRISPR can copy itself to the other chromosome, meaning almost all of the parent's offspring are going to inherit that gene. This means that entire species can be eliminated in very little time. If we return to the mosquitoes example, we have the ability to wipe out mosquitoes very quickly. The problem with that is mosquitoes are pollinators. They also make up a part of the food chain. So without mosquitoes, there can be a lot of unintended effects. We can have we can have a decrease in other animals because they're losing their primary food source. We're losing pollination of a lot of plants. So in general, I don't think it's ever a good idea to make something go extinct. But with CRISPR, we do now have that ability. On the flip side, reintroducing a species also has its problems. It can disturb the ecosystem a lot of ways. Depending on what you introduce, it'll introduce competition for food and land. It can increase or decrease the ratio to predators and prey. They can also become new hosts for disease because they haven't developed immunity to modern viruses the way other animals have. Which means they're more vulnerable to those, and if those diseases mutate, they can then be passed on to the original animals that originally had immunity. This is something you can see returning to the woolly mammoth example. Am I out of time?
You did give a couple more minutes to wrap up.
Okay, sounds good. I think this is my last one.
Okay.
If we were to introduce the woolly mammoth per se, Not only would it be competing with other elephants for food and resources, but it would also be responsible for a lot of the destruction of undergrowth, and just part of it's the habitat that it was living in. Another problem with reintroducing species is that it's going to introduce kind of an apathy towards the environment. The problem, when animals go extinct, the problem isn't just that a singular species has just gone extinct. It's that humans don't always treat the environment the way we should. Oceans get polluted, forests get chopped down, garbage starts piling up, and I don't think it's very prudent to introduce a new species into a world where current species are already struggling. And I think it'll also increase the apathy towards any endangered species because people are going to view them as replaceable. There's no point worrying about a species going extinct if you can always bring it back.
Oh my gosh, I'm so sorry. I did not realize that was happening.
So what should we take from this? CRISPR is a very powerful tool, and it is capable of a lot. But it's not something that should be taken lightly, and discretion is needed to determine what should be modified and what shouldn't. So I hope that in the future, CRISPR will be carefully regulated to prevent any disasters from occurring. And I hope that I've convinced y'all that CRISPR is something to watch, both for its abilities but also for its risks. So that's all.